we've talked about 3 out of the 4 organic macromolecules or biological molecules. We've talked about carbohydrates, lipids, proteins. The last one that we need to talk about are the nucleic acids. So we're going to just sort of talk about the basics of nucleic acids. In this unit, we're going to briefly mention protein synthesis, but then we'll talk about it more when we get to our genetics unit. So nucleic acid is the polymer molecule. And nucleic acids are really important to living things. They make up the genetic code for all living things. They also allow for that genetic code to be transformed into proteins, or to be used to make proteins. There's 2 main types of nucleic acids. We've heard of both of them, DNA and RNA. DNA, the letters stand for deoxyribonucleic acid.
We don't have to memorize that, correct?
You should recognize it. And so, can you guess what RNA stands for?
No.
Oh wait.
Reox.
Yeah, ribonucleic acid. It's just missing the deoxy part. So slightly different molecule.
Why is it an acid? Is it low pH?
No, there's depending on the groups that are found, the side chains and things like that determine whether something's considered an acid. So DNA is found in the nucleus of cells that have a nucleus. It's also, as we talked about, there's some DNA in the chloroplast, there's some DNA in the mitochondria, and even other organelles as well.
Is this a generalization, or is that actually, if we could...
Like if what it looks like?
Yeah.
So, yeah. So this is, you mean this diagram on the lower right?
Yeah.
Yeah, we'll talk about the shape in a couple minutes. So DNA, like the other organic macromolecules, are made of hydrogen, carbon, oxygen. Also nitrogen and phosphorus are present. It's a very large polymer. So it's a polymer, meaning it's made of many, many repeating monomer units. The monomer of a nucleotide of a nucleic acid is a nucleotide. Okay. This is one nucleotide. The building blocks of the nucleic acid. A nucleotide has 3 parts to it. There's a sugar, there's a phosphate, and then there's a variable group we call the nitrogenous base. The sugar, it's a 5-carbon sugar, deoxyribose. We can recognize it here because of that ring shape. The phosphate group is a phosphorus atom bonded to oxygen. And then there's the nitrogenous base, which is attached to the sugar. So if you think about the amino acids, how many possible side groups could we put onto a amino acid backbone? There were 4 things attached, but that R group, how many different R groups were there?
6.
Somebody said over here. 20. There's 20 different things. That's why there's 20 amino acids. For DNA, there's only 4. 4 possible bases can be attached. They're adenine, thymine, guanine, and cytosine. A, T, G, and C. We often symbolize them. So this is one monomer. And if we link together, many of those will form the polymer, which is a nucleic acid. This is showing a DNA molecule. The shape of the DNA molecule is very distinctive. Okay, it's called, what's the name of it? Yeah, it's called the double helix. It's sometimes referred to as like if you took a ladder and twisted it, So that would be the structure of the DNA molecule. Or if you think of a spiral staircase, that's the shape of the DNA molecule. It's double-stranded, so there's two sides to a DNA molecule. The structure of DNA was discovered by James Watson and Francis Crick in the 1950s. Although it had some help from a female scientist who really did not get the credit that she deserved. Her name was Rosalind Franklin. She basically took X-ray images of molecules, and she took it, it's called X-ray crystallography, and she took an image of a DNA molecule that Watson and Crick saw, and it prompted them to sort of change their model and make it the double helix, which it turned out to actually be. She did not get the credit that she deserved. And how they obtained her X-ray crystallography image was sort of controversial. She didn't give that to them, share it with them. Somebody else saw it and told them about it. So it's an interesting story. I think we'll talk more about it in genetics. So if we look at a model of DNA here, the bases in DNA are what we call complementary. They match up in certain patterns. Adenine, if adenine is on one strand, its opposing strand will have thymine as its base. A and T go together. If one side has guanine, the other side will have cytosine. You say that those bases are complementary. And that's important because when a DNA molecule, like this, needs to be copied. So every time a cell divides, it needs to completely copy all of its DNA to provide each cell with a copy. So that's important. And because of those complementary bases, the DNA molecule can split in half down the middle into two strands, like this. And then nucleotides that are sort of floating around in that area can be attached by enzymes matching up properly. So T's will attach to A's, G's with C's, and so forth. And after that happens for the entire DNA molecule, what you're left with are two identical copies. Okay? The dark blue represents the original strands. They've been filled in with complementary bases, and now we have two copies of the DNA that are exact copies of each other. This is how DNA replicates itself. If we look a little bit more closely at the bases, right, they come in two categories. Pyrimidines, which are these single ring structures. Thymine is a pyrimidine. Cytosine is a pyrimidine. Or these double ring structures, which are adenine and guanine. They're called purines. And they are complementary. A and T, as you know, match together. C and G match together. So a purine and a pyrimidine match up with each other. And we can see that here, where we have T and A, C and G bonded together. Now, holding the two separate strands of the DNA molecule together are a bond we've talked about early in this unit, hydrogen bonds. Somebody tell me something about hydrogen bonds that we learned about. I think it was one of the first things we did. Can you tell me about hydrogen bonds? Anybody? David?
They only have one bond.
Well, hydrogen can form a covalent bond, but these aren't covalent bonds.
Oh,
We've learned about these talking about a specific important molecule that forms hydrogen bonds between the molecules, and it gives it cohesive properties. What molecule forms hydrogen bonds with itself? Water. You remember the first pack we did about water? Water forms hydrogen bonds between, remember you were drawing the molecules? You had to match them up properly so that oxygen and hydrogen form those bonds together. Hydrogen bonds are weak bonds. They're not that strong. Why do they form? You think back to that water, why did hydrogen bonds form between the molecules. Something about how the water molecule is structured. Remember back to the diagram, one part was shaded more than the other part?
What was the electrons?
Yeah, the electron density. Where were the electrons more dense?
Around the oxygen.
Around the oxygen. What did that give the oxygen?
Negative charge.
A negative charge. And what did the hydrogens have?
Positive charge.
A slight positive charge. And therefore the water molecules were attracted to each other. That same concept applies to these bases. Okay? Part of these bases, the hydrogen, becomes slightly positively charged, which attracts it to a slight negative charge on the opposing complementary base, and that forms hydrogen bonds between them. Now, individual hydrogen bonds are relatively weak, but when they form one after another at every base pairing, they can give the DNA molecule stability and strength. But they can also be separated relatively easily so that the DNA can replicate in this way. Mateo?
Why is there 3 bonds for CG and only 2 for TA?
Where are we looking? Oh, it's just because of the structure of these molecules.
Oh, okay.
Yeah, about how those hydrogen bonds form. All right. There's another nucleic acid, however, besides DNA, that's important in living organisms, RNA. RNA is found in the nucleolus. It's found in the ribosomes and also through the cytoplasm. And RNA is, again, it's a nucleic acid. It's a large polymer. However, it's only single-stranded, not a double helix. It's a single helix. It's also made of nucleotides, which have a sugar, a phosphate, and a base. So that's the same. In RNA, the bases are cytosine, guanine, adenine, but they don't actually have thymine. They have a substitute. RNA has a base called uracil instead of thymine. Just those. Yeah. It also has a different sugar. What was the sugar found in DNA? What does the D stand for? Deoxyribose. In RNA, it's just ribose. It's ribonucleic acid. And RNA is important. It helps produce proteins. We'll talk a lot about that in the next couple slides, but not today. Any questions about the nucleotides? All right. So you have about 5 or 6 minutes.